Hi, so today we're going to talk about neurophysiology, so the brain, nervous system. We're going to talk about central nervous system structure and function, and primarily we're going to focus on structures and functions of some of the things in the brain, and we're also going to have an intro to that before we discuss those things. So just to get oriented again, the central nervous system is the brain and spinal cord. Peripheral nervous system is everything outside the central nervous system. And so we're going to have separate sections for sensory and motor. All right, so let's look at cells of the nervous system. We've already kind of talked about neurons and how neurons release neurotransmitter. They have the action potential that allows them to communicate with other either effector organs or other neurons. So receive stimuli and transmit action potentials. They have the neurotransmitters, and then the neurotransmitters are what can have that effect. Okay, so we kind of went through that. We're going to now talk about neuroglia. So initially when people looked at the brain with their little microscopes long ago and tried to figure out what was going on in the brain, they could see the neurons, and they kind of figured out that the neurons had this communication function, but there were these other cells that were kind of in there, kind of holding, it seemed like they were just holding things together. So glia means glue. So they were the nerve cells that were kind of the glue holding things together. And they do that, they do offer support and protection, but they also serve specific roles. So we're going to talk about those. So parts of the neuron, we've been over this kind of unofficially, but this is just kind of officially going over what the what a neuron is. And it has, just like any cell, it has a nucleus, and that nucleus is contained in the neuron cell body here. And the nucleus does the same thing that it does in other cells, and that is it holds the DNA. And then the rest of the cell body has the Golgi apparatus, the rough endoplasmic reticulum. Golgi, as remember, puts off those vesicles and allows for exocytosis. The rough ER is where you have those little ribosomes that are pumping out DNA, putting those amino acids together. So it does the same thing, that protein synthesis. But then those proteins, and really we talk about neuropeptides that are produced, and even though we have this long axon, the neuropeptides and the other proteins too, all of the proteins have to be made up here in this cell body. And then they have to travel down the axon, and that doesn't matter whether it's a very short, like an interneuron, those are very short, or long, like a meter long, because it goes all the way from your spinal cord down to your feet, to your toes. Okay, so we call that movement, we call it axoplasmic transport, which is where something kind of walks along. So microtubules are part of the cytoskeleton. So we have these kinesin molecules that kind of have these foot processes, and then they can attach to something, and then they will carry it down. They will just kind of walk down these microtubules and carry proteins, neurotransmitters, mitochondria. This looks like a mitochondria here. And they can move down, and then they can also remove waste products from the other way and bring those back to the nucleus. So it can be fast, slow, or it can be anterograde, which means away from, if the cell body's over here and into the neurons here, then it's going to be moving. Anterograde is toward the synaptic terminal, and retrograde means it's going back to the cell body. Okay. Other things that should be familiar by now, hopefully, are the dendrites, and that's where the input towards the cell, the neuron, happens. And then those dendrites have little spines on them. You can't really see them in that picture, but they have little spines growing off of them that kind of add surface area or places for incoming neurons to pass their signal on. Okay, so let's see. Axons. The axon is that long part of the cell that transmits the action potential. And the very beginning of the axon is this area called the axon hillock. So when we have a depolarization event, so if we have a nerve that kind of, or a neuron that synapses up here, then it'll pass a signal down, and then if we have enough of those, okay, then we may get that summation, and it may make it down. If it makes it, if the depolarization or like that would be excitatory or inhibitory would stop it. But if you have an excitatory signal that makes it all the way down to this area called the axon hillock, then you'll have a complete depolarization event, okay, or a complete action potential. Now, so really what's happening here is that you're causing depolarization of the cell body. That's what we call a graded potential. It gets down here to this axon hillock, and there are a bunch of voltage-gated sodium channels, just like there are kind of all the way along here, or at least in the nodes, that's where they are. But when it hits those first, let's see, draw that there. When it hits those first voltage-gated sodium channels and the sodium rushes in, then it's going to cause more depolarization, and then the whole thing is going to just go as an action potential. Okay, so the sodium rushing in, depolarization, potassium rushing out, repolarization. Okay, so that's that initial segment. This whole area is kind of the initiation or the trigger zone, and that's where the whole process begins. So site where action potentials are generated, axon hillock, and part of the axon nearest the cell body. Okay, so that's the trigger zone, that area. And then we move down here to the terminal, so the axon terminal. And if it's synapsing with something else, then it's presynaptic, and then you have the synapse, and then you have the next neuron or muscle or gland or whatever it is that it's synapsing with. Okay, and then of course the synaptic vesicles is what holds the neurotransmitter at that terminal. So types of neurons, functional classification. We talk about really whether it's going to the brain or away from the brain. Okay, so sensory is going to the brain. We call that afferent. So that's afferent signaling, goes towards the brain or towards the central nervous system. So that includes going towards the spinal cord, and which is going to also go up to the brain, unless it's a reflex. And it's going to be, so sensory is going to be afferent. Motor is going away from the central nervous system, so away from the spinal cord, and that's efferent. Okay, so those are the terms, just the terms we use for that. And then interneurons are kind of in between. They're exactly what it sounds like. So if you have a sensory receptor, so you have something over here, you touched something, you've maybe felt pain, it moves towards the central nervous system, and then you'll have these interneurons, these short interneurons that will relay that message, and then you may have an efferent signal back to the muscle to tell the muscle what to do. So this could be a relay in the brain, like a reflex type of thing, or a reflex in the spinal cord, or it could all go all the way up to the brain. Okay, it's just those interneurons are just short and unmyelinated, and usually these longer tracts are myelinated. All right, and then structural classification. So we have multipolar, which includes most neurons, and they just have more projections out from the cell body. So if you're trying to figure out whether it's multipolar, bipolar, or unipolar, multi means many. So you just, in every case, look at the cell body and say how many projections are coming out from the cell body. Well, there are multiple projections coming out of the cell body in this one. So one axon and then a whole bunch of dendrites. This one, there are only two projections coming out, so we call that a bipolar neuron. And this, there's your cell body, one projection coming out, so we call that a unipolar. Okay? So this is just that classifications. If you look at these microscopically, those are the three types that you'll see. And usually, like I said, most of them are multipolar, but there are some of these bipolar and the unipolar in specific places. All right, so organization of nervous tissue. We often hear about gray matter. Gray matter just means unmyelinated. So That includes the axons, unmyelinated axons, cell bodies, dendrites, neuroglia, really anything that's not myelinated, that's in the brain and is a neuron of some sort or neuronal cell, is considered gray matter. Okay. White matter is myelinated. That's what causes it to be white, and that's really it. So it's white because it's myelinated, and if you have a myelinated neuron, it's usually because it's a tract that's trying to communicate at some kind of distance away, and so we want faster. Remember, it kind of uses that current flow, and then it kind of jumps node to node very quickly. And that's what we're after with myelinated. So they work well for kind of distance communication. All right? So if we think about that, if we think, okay, myelinated are for tracts that are communicating over a distance, then we think of the spinal cord, and that's really a lot of what's going on in the spinal cord, is we have neurons going up and down the spinal cord to get out to, you know, the legs, the digestive system, the arms, the, you know, everything all along the way are going to be distance type of communication from the brain. So it's taking those messages from the brain, and then they will synapse in the spinal cord, and then you'll have neurons that go out from the spinal cord. Okay, so we have these signals going in, and then there's a synapse in that gray matter area, and then these will be myelinated as they leave. But in here you have a bunch of interneurons communication that causes that gray matter to kind of be on the inside. Same thing's going on in the brain, except you can see it's kind of reversed. Here in the spinal cord, we can see mostly the outer part is myelinated, and then in the brain, the outer part is unmyelinated, and the inner part is where the tracts are. So communication is kind of coming in. Okay, let's say that's the thalamus. So we have communication going here, and then the process is happening, or the processing of information and memory and things like that are kind of happening out here on the outer part of the brain. Okay? So synapses may be happening here from information coming in. Okay? And so you have some of these short interneurons. And really, where you have cell bodies collected, we call that a nucleus in the central nervous system. And that's kind of what you're seeing. That's the gray matter. There could be some interneurons here, or just some synapses with some cell bodies in that area. So that's gray matter. And then out here you have a lot of kind of structured, organized communication, small neurons that are synapsing with other smaller neurons in that area. And like I said, that's memory processing, and we'll see some of those areas, like when we talk about the primary motor cortex or the primary sensory cortex, we'll see some of that. So in the brain, gray is the outer cortex as well as the inner nuclei. White is deeper. In the spinal cord, white is the outer, gray is deeper. And the peripheral nervous system, gray matter is groups of cell bodies called ganglia. In the central nervous system in the brain, they're called nuclei. And by that I mean just we have synapses, we have cell bodies that then can communicate. And where those nuclei, so if we have a bunch of those, where those nuclei are kind of gathered, that's what we're talking about with, the cell bodies are gathered. That's kind of what we call the nuclei and the ganglia. Okay, so it's just a term that you'll see, and it's just good to know that that's, you know, one of the things that we're talking about there. Okay, let's talk about specifics. Let's talk about glial cells. Glial cells, remember that was the glue that we initially thought was the glue, and if you look at this, if you consider, well, these are the neurons, these guys here, and these are the workhorses doing the stuff, and then you see these other cells just kind of hanging out on the side, maybe having some contact, like this guy, this astrocyte is kind of holding on to that blood vessel, and it's holding on to there. It almost looks like that's kind of holding things together, and it does do that. But there's a lot going on. We'll talk about the astrocyte. That's where the blood-brain barrier is. These cells, the epidermal cells, these are producing cerebral spinal fluid. Oligodendrocytes, these guys, even though they've got these little foot processes, but what they're doing is they're actually producing myelin, and they're creating those myelin sheaths for that faster current flow under the neurons there. Microglia, these guys are running around kind of cleaning things up. They're kind of like a little bit like macrophages maybe in the immune system. They're kind of cleaning up cell debris, but they also have an immune function similar to the immune system in the periphery. Okay, so they're kind of the central nervous system's immune system here. Okay, so astrocytes, again, that's everything I just said. Support cells control extracellular environment of neurons, and they are that blood-brain barrier. Microglia, immune system, and they remove that debris. Oligodendrocytes responsible for myelin in the CNS, and Schwann cells are responsible for myelin in the peripheral nervous system. And then the ependymal cells are ciliated, so that means they're kind of pushing along the cerebral spinal fluid. Okay, involved with the production of CSF and CSF movement, cerebral spinal fluid. So unlike neurons, glial cells are able to divide, which kind of is important for, well, a lot of things, for regeneration for one thing, but when that goes wrong, if you hear about cancers, a lot of times you're talking about a glioma, or like an astrocytoma or a glioblastoma, which are astrocytes when astrocytes become cancerous, and these are the most common. Others will too, and you could get cancers, I believe, of like neurons, but that they're going to have to be triggered to grow because neurons aren't typically mitotic; they don't divide. And so that's what happens with cancer, is you have out of control cell division. So the fact that these can divide kind of makes that makes them susceptible, I guess, to cancer formation. Okay, so this is just a slide that kind of goes through all of these and tells a little more information about them and has some nice pictures, so I left it up here, but I kind of divided those out in the next few slides. So let's talk about the astrocytes. That is the site of the blood-brain barrier. So we can the blood-brain barrier. So the brain has its own fluid. Here's the cerebral spinal fluid, but so blood vessels, there's a lot of vascularization in the brain, but for fluid and proteins to move out of that vascular area, to move out of the blood vessels and to be fluid kind of bathing the cells in the brain, it has to kind of go through this check. So the purpose of the blood-brain barrier is to protect neurons from toxic substances, and toxic could mean anything. I mean, toxic may mean toxic to the brain, even though it's fine in the periphery. Allows the exchange of nutrients and waste products between neurons and blood, prevents fluctuations in the composition of blood from affecting the functions of the brain. So water is able to move through aquaporins pretty freely. So the water inside the blood vessels can move in and out, okay, if it has to, which is, which is good, obviously, or it wouldn't be that way. But in times of like dehydration or, high salt, low salt, or rapid changes, that can actually cause problems because that fluid is able to flow. So just remember that water is able to penetrate easily. It can go in and out of the blood-brain barrier. It's the proteins and other structures that aren't able to pass through. Okay? And those are the ones that have to move physically through these astrocytes. So we have down here, they don't pass between the cells. So substances, we've got these tight junction here. It won't let any of these, so if there's a protein in here that wants to get into the brain, it has to go through that process, through that foot process of the astrocyte, and then it can move through the cell. Okay, so they have to pass through the cell, makes it very selective, and this is something to be considered when developing drugs. So if things are a little more hydrophobic, then a lot of times, or lipid soluble, a lot of times those can pass through a little more readily, but otherwise it's pretty specific as to what it lets through. All right, so Schwann cells and myelin, we've mentioned the oligodendrocytes. Oligodendrocytes, they produce the myelin in the central nervous system, and Schwann cells produce it in the peripheral nervous system. Myelin in general increases the speed of an action potential by increasing current flow. I want you to understand what that means when I keep saying it increases current flow. So it insulates that neuron. I don't want to spend too much time talking about this, But when you have a depolarization event, when sodium depolarizes, because you've got sodium depolarizing, rushes in, depolarizes, potassium rushes out, repolarizes, it happens very, very quickly, like, 100 times per second. But overall, in the overall scheme of things, that's slower than if we could just like make an arc and just send it down. Okay, but that's what happens with the myelin. The myelin kind of insulates it, so you can have that current flow. So when you have the depolarization event, that depolarization event will just follow it down and just go straight down. You don't need to depolarize, repolarize all the way down. You can just have sodium rushing electrically down that depolarization rush down here, and then it'll move to this point where you may have more voltage gated channels. Okay, so it increases that current flow. As a review, okay, so myelinated axons are about 100 meters per second, depending on the diameter of it, and there are a lot of things that depends on, but just in general, they're maybe 100 times faster than an unmyelinated neuron. So unmyelinated, 1 meter per second. And if you consider that a neuron going from your spine to your foot if that's about a meter, then a down and back trip is going to take 2 seconds, and that's not fast enough. It needs to be faster than that to pass information like muscle control or proprioception, which we'll talk about. Oligodendrocytes and the myelin, we already said that. So degeneration of myelin sheaths occurs in multiple sclerosis, in some cases of diabetes mellitus, and there are other demyelinating diseases also that you'll learn about hopefully in patho. But you can kind of predict the effects, weakness, uncoordination, that kind of thing happens when you start to have demyelination of the neurons. Okay, so let's talk about structure. So central nervous system, the brain, so that includes the spinal cord, so we can kind of get that out of the way, and then we're going to kind of work our way up through the brain. So the spinal cord is down here, and it has projections that go out between the vertebrae as spinal nerves. Okay, and they go to the periphery. So those are peripheral because they're leaving the spinal cord, or they could be afferent and coming in. Okay, but the tracks going up and down the spinal cord are considered part of the central nervous system. And then we'll kind of move up to the brainstem, where we have the medulla and the pons and the midbrain right here. Okay, and then we'll have the forebrain, thalamus, hypothalamus, cerebrum. We'll get more into that. And then the outer part of the brain is the cerebellum. Or I'm sorry, the outer part of the brain is the cerebrum, the back part of the brain, that's the cerebellum. Okay. All right, so cerebrum, I said we're going to start at the spinal cord, and we will, but first I want to talk about some of the regions, the overall, I guess, looking at the brain and seeing how it's sectioned off. It's composed of right and left hemispheres, each of which has the following lobes: the frontal lobe, parietal lobe is kind of in this area. This is something you'll cover in anatomy, I hope. Occipital lobe, which is back here in the back, temporal lobe, and then the insula is kind of buried, or temporal lobe, and then the insula is kind of buried under the, so there's temporal lobe here. The insula is kind of an island. It's kind of under, if you pull away that temporal lobe, you can see the insula. Okay. So those areas, and then functionally, which is what we're really more interested in, we have these primary areas. The primary motor cortex, we'll talk about that for muscle control and processing, and this is where the muscle is processed and said, here we go, we're going to move this arm, this leg, whatever. That all starts here, or that's all processed there to send the signal. It's communicating with a lot of other places and to get to that point. Same with the sensory, primary somatic sensory cortex. Same thing, but now with sensory information, it arrives there and then it's processed. We also have the visual cortex back here, where vision first arrives after, you know, it checks off in the thalamus, but then it goes back to that visual cortex, and that's where it's initially processed, and then you add things together like depth and motion and things like that. Auditory cortex, same thing. It's kind of in the temporal lobe here, and that's where sound is processed. Okay. The frontal lobe, I want to say more about that because that's the only lobe function that I'll ask any kind of questions about. So the frontal lobe is what it gives us our ability to plan. So the frontal lobe, if you look here, the frontal lobe of, that's not colored in the rat, but this is, or the cat, but this is the frontal lobe of a cat. Well, kind of this area, yeah, which is a little less developed, and then the rat, it's even more or less developed. But in humans, it's very well developed, and that kind of is what separates us from other species, because of that. It's looking ahead. It's kind of playing something out before it happens. I heard somebody give an example of, you know, Ben & Jerry's never made liver and onions ice cream because they could kind of play that out in their prefrontal cortex and decide, okay, that's probably not a good idea. We use the prefrontal cortex for things like, you know, we know that we have to take a lunch if we're going to be gone all day. We kind of can see the future in a way. You know, we're getting 401ks and going to school and that kind of thing. For hunting, it's good for like animals like cats and dogs and monkeys, because they can kind of predict what their prey is going to do and when to find the prey, where to find the prey. They can kind of predict what's going to happen. But it's developed very well, almost to a fault, in humans. And that prefrontal cortex is, many psychiatric disorders affect that frontal lobe, or affect frontal lobe communication. So we've got a lot of power with our frontal lobe in sort of seeing the world and predicting what's going to happen in the world. But imbalances in that can cause psychiatric disorders too. Okay. All right, so now we're going to start from the brainstem. We're going to start with the medulla oblongata, and that is the most inferior part. So you can see the spinal cord just kind of has this nice smooth transition into the brain, and that's what the medulla is. It's continuous with that spinal cord, has both ascending and descending nerve tracts. Gray matter, so this is the short neurons that are unmyelinated, that are just sort of there for processing, to use kind of a generic term. They'll get information, and then they can process, like if your heart rate is slow or fast, then they can kind of process that information and then send a signal to speed up the heart or slow down the heart. Same with blood vessel diameter, respiration, swallowing, vomiting, hiccuping, coughing, and sneezing. Those are all kind of reflexes that can be found in the medulla. So a lot of times we talk about primitive function. So this is something that has to function in every species. And so the brainstem is very similar in every species. And so when we talk about life, and you know, you may contain brainstem function, which means that you can still regulate your heart rate and blood pressure to some degree, but you know, a lot of times you may lose function of the cortex. Okay. All right. So some other things that are found in the medulla, something called pyramids, and pyramids are shaped like pyramids, and really they're just motor nerve tracts. Okay, so your motor cortex is kind of up here. Move forward. So, and then they will send a signal down to this area, and they all kind of go through this same zone, and it's shaped like a pyramid, and so they call it pyramidal. But you also have information. So that's like your gross motor movements. I'm going to move this leg, and I'm going to move it that far, or whatever. You have another area here. This calls it the olive, but we're just going to think of it as being extrapyramidal, outside that pyramidal system. So if we I know this is a really small picture, but if we have the pyramidal tracts here, then we may have something coming from the cerebellum here. You may have other stuff coming from the, it doesn't stop and turn around like I just did, but you may have something coming from other brain regions, like for balance, your vestibular system may get some input, your vision may get some input, and those are extrapyramidal. They aren't directly controlling muscles, but they can tweak the muscle contractions and some of the other muscles around it to give balance and coordination. Okay, so the pyramids are really the motor function. The extrapyramidal, which is the olive, extrapyramidal, which means outside that pyramidal area, are for balance, coordination, stuff like that. Okay, and we'll come back and talk about that again when we talk about motor function. All right, so let's move up. Oh, and also 10 of 12 cranial nerves exit through the brain stem in general, the pons and medulla. Okay, so now let's move up from the medulla to the pons. It's superior or rostral towards the nose from the medulla, and it also has these ascending and descending tracts, and they relays the information between the cerebrum and the cerebellum. So we'll talk about the cerebrum, but a lot of communication happens between there, and it kind of moves through that pons region. Okay, the respiratory center. So we had that in the medulla as well, but we have the respiratory center has two parts: the depth and the rate. And one of those is in the medulla, and one of those is in the pons. You're in the pons. Midbrain. So if we go up a little further, we get to the midbrain. Midbrain has a lot to do with, it's a small area, and it's pretty specialized. It's small and superior to the pons, but it's a busy area. So there are two superior and inferior colliculi involved in visual reflexes, so being able to maintain a straight ahead gaze even though your head is moving. So you've got communication with, again, your vestibular system, muscle control, and it's kind of monitoring that, so you can automatically kind of keep your eyes fixed on a point even though your head is moving, and that's something you don't have to think about. So that's kind of going on in there. Hearing. So if you think of hearing a sound, let's say there's a sound over here. This is a speaker that's emitting sound, and you have one ear there and one ear there. That sound is first going to arrive at this ear, and it's going to start moving, and then the sound is going to make it, this is your head in the way here, it's going to make it to this ear, but it's going to be a little bit later. Okay, so what happens is, because it got to this ear first, it's going to be a little bit off center. Okay, so when it hits that area, you're going to see that they kind of meet up a little bit on this side. And what that means is that the signal reached this ear first before this one, because it didn't have as much time to travel over here. And even though this is very, very small, very minor differences, that's kind of how we locate whether sounds are left or right. We also can take into account the volume, but you also have this automatic kind of, your brain automatically kind of can tell by timing it where the sound came from, which is pretty amazing. Something else that's significant here is there's a little area called the substantia nigra. Nigra meaning black. It's pigmented with melanin, and that plays a role in reward and addiction and really movement. Okay, so I've mentioned before Parkinson's disease, and Parkinson's disease is a disease that's caused by, it's a motor disorder, and it's associated with tremor, not being able to initiate movement. So you're you can kind of be frozen a little bit, and you have a hard time taking those first steps, and that's because the substantia nigra, this brain region, kind of goes away over time. So they're dopaminergic cells in there, and they kind of die over time. in everybody, but when you lose, I think it's like 80% of them, then you start to show these symptoms of Parkinson's disease. You start to have these motor symptoms. And, you know, usually when we see people with Parkinson's and their movements are because of, you know, additional drugs, and it's hard to get the medications right. But that's the area. So these dopaminergic, so dopamine neurons in the substantia nigra die, and that's what causes Parkinson's disease. Okay, so dopamine neurons in the substantia nigra are destroyed in Parkinson's disease, causing the symptoms. Some treatments activate dopamine receptors and cause compulsive gambling. I don't want to get into that because that's more of a trivia kind of a thing. But what we're not trivia, but it's not relevant for what we're doing right now. But the thing that it does kind of allude to is that we're trying to increase dopamine in that area. So we've got a loss of neurons, and the treatment of choice is L-DOPA. Okay, so L-DOPA is the precursor to dopamine, crosses the blood-brain barrier, moves into the brain, is converted to dopamine, kind of increases your dopamine levels, which allows the patient to regain some function. But there are also dopamine agonists that may work. And then if we ever got into detail on it, like in patho, or pharmacology, you may talk about like some acetylcholine antagonists, stuff like that. But mainly we use L-DOPA, but some of these agonists can cause kind of funky side effects. And that's what this is saying, is that you got to be careful with the patients, because different patients will handle these agonists differently, and some of them can cause compulsive gambling in them. Okay, let's see. I said a lot about that for not wanting to say anything about it, huh? All right, so moving up again to well, we're kind of jumping over the thalamus, but we're going to move to the thalamus, and that is the major hub of sensory information. I want you to understand the importance of the thalamus and really how it sits there and what's going on with it. It's kind of interesting. So sensory information from the spinal cord and the cranial nerves, information that's coming in, will synapse in the thalamus. Okay, and so like with vision, and it'll show that in the next picture. Well, let's use one of these. Sensory integration, motor function, ventral anterior lateral nuclei. So information will come in, synapse, and then be processed wherever it is. So like we're going to learn about sensory very soon. Sensory information comes in, it gets to the thalamus, and then the thalamus will direct another neuron to the sensory cortex, or the motor cortex may move through here, and well, it actually moves directly down, but you can kind of get the idea that you've got sensory information moving in, synapses, and then moves off to a different area. And so the thalamus kind of directs where everything needs to go. Okay, so it moves through that area. That way you don't just have everything just going all over the place. Everything kind of goes to this central location. Well, I made a mess of that. See, that's what it would look like without a thalamus. So it goes to the thalamus, and then the thalamus is able to direct those tracks to where they need to go. Okay. So apparently that's a much more efficient way to do things, because that's how it's done. And the body's efficient. All right. So these are just some examples of information that is processed in which part of the thalamus and then relayed to a different location. Okay, and another picture of the same thing. So this is visual, so this is the optic nerve, optic chiasm. Part of that information will go left field versus right field. Will go to either the left or the right thalamus. There are two of them, and then that will be processed and sent back here to the visual cortex. Okay? And it maintains the fidelity of the information that's coming here. Okay, so I don't want to jump in too much. So if you've got something coming in from this side, it's mainly processed over here, then that information actually won't cross over, and then it'll be processed over here on that side. Okay, but it has to go through that thalamus. All right, and then we have the hypothalamus. So feedback for overall body condition. We kind of talked about this when we talked about autonomic innervation, the autonomic nervous system. So feedback for overall body condition eventually makes its way to the hypothalamus, even if you aren't aware. So some things listed down here like temperature regulation. So we maintain our body temperature by maybe controlling sweating or glucose burning. Hunger and thirst. Thirst leads to direct regulation of salts and water. So what happens in general is that, and as we go along, we're going to see that the hypothalamus is going to cause the release of various hormones from the pituitary gland. I mean, we've talked about, or we haven't talked about them, but you may be familiar with some of the hormones, sex hormones that are released from the pituitary gland. But there's more. There are more than just that. There are hormones that act on the kidney, the thyroid, and a lot of that is tuned. So information comes into the hypothalamus, and then the hypothalamus responds by telling the pituitary gland, which I keep trying to move forward, which is next, which hormones to release. Okay, so the main way the hypothalamus controls conditions in the periphery, it's through that pituitary. So it activates the pituitary gland, tells the pituitary gland which hormones to release. Those hormones are then released into the blood, and then they'll have an effect on whatever organ system or functionality that needs to take place. So hormones affect many things that we'll talk about, and that hypothalamic-pituitary axis is wherever it goes next is a link between the brain and the periphery. So we have brain things going on. You can tell yourself to be stressed out just by thinking of something. The hypothalamus sends a signal to the pituitary gland. Pituitary gland releases cortisol. Well, it releases, let's see, I think corticotropin releasing hormone. Anyway, it can cause release of cortisol from the adrenal gland. And so now you've got it, and now you've got your whole periphery is affected too. Okay, so it's a great process. Just by thinking about something, you can get stressed out. That's the joy and bonus of physiology. All right, so cerebral medulla. So we're kind of moving up. So we had our thalamus, hypothalamus, and pituitary, and now we have this area called the cerebral medulla as opposed to the cerebral cortex. So the cerebral medulla is the white matter between the cortex and some of these inner structures, between the cortex and some of these inner structures. We also have the corpus callosum. Corpus callosum is a tract that kind of goes laterally. It's connecting the left and the right halves. Maybe you've heard about people with severe forms of epilepsy that have that cut, that cerebral cortex cut, and if that happens, when they first did that, there weren't obvious signs that there were changes, but then if you had something, I kind of drew a little bit how if something is coming in from your right field of view and it goes into your left side of your brain, that your left side without that corpus callosum to share that information across that tract won't be able to share it with the right side. Okay, but yeah, so it's left-right communication. That's what the corpus callosum does. And so you can kind of see that here, the lateral, and then you can kind of see how these fibers, these axons, are communicating also into different areas. Okay. And really a lot of this communication is like with the basal ganglia, between structures of the basal ganglia, which we'll talk about in I think 2 slides. And so yeah, just a bunch of tracks like that. All right, so the last structure we're going to talk about is the cerebellum. And I have here, it says the cerebellum is well connected, and that's because the cerebellum kind of works as a separate little brain. It's got a lot more short, non-myelinated neurons in it, but it definitely sends out processes to the entire brain, and that's what this is showing. I didn't point that out, but this is just a kind of a map of axons and how they communicate and where they're communicating. And you can see that a lot of signaling will stop from that pons area, will move from the cerebellum to the rest of the body. So kind of out through that area, and it kind of communicates with every part of the body. So so it's involved in locomotion, balance, and posture, but that kind of doesn't give it justice, because it's so your parietal lobe kind of gives you the spatial recognition without like looking all over the place. You can kind of tell where things are in space around you, and so you may have communication there. So let's imagine the good example here of riding a bicycle. You have to kind of sense a lot of things. You have to have your balance, you have to have spatial awareness, and then you have to have coordination of your feet making everything move. And so the cerebellum, once it is trained, so it's difficult to learn to ride a bicycle. If you remember learning, or if you've ever watched a kid try to learn to ride a bicycle, you're thinking about it too much. You're trying to use your cortex, your cerebrum, but you're training this cerebellum, and then the cerebellum is able to just kind of take over and do that without thinking about it. Then you can have a conversation, and you don't even pay attention to what you're doing anymore. So that's a lot of the function of that region. Okay, so of that structure, the cerebellum. Oh, and I have here cerebellar cortex has 10 to the 12th neurons, which is a whole bunch, but it's smaller than the cerebral cortex, or which is more, not smaller. It is smaller than the cerebrum, but it has more neurons than the cerebrum. Okay, but it can't think of things. It can't think of, what you want to have for lunch. At least I don't think it can. All right, so the basal ganglia, okay, a lot of times it's called the basal ganglia, but technically, because they're structures inside the central nervous system, we're supposed to call it the basal nuclei. But this area is an assortment, or a group of structures that work together for motor function control. Okay, so planning will take place kind of in the prefrontal cortex here, the motor cortex. But there's a lot of communication between different structures to provide balance and finesse, I guess, purpose, continued movement, that kind of thing. So there are a lot of different structures involved with the basal nuclei. We're going to talk about some of those when we talk about motor control. Okay, motor function. But basal ganglia, just know right now, I mean, it's all going to be on the same exam, but right now we're just kind of introducing it as a term. All right, limbic system. This is involved in emotion, so collection of structures involved in emotions and memory. It has a lot of parts. It's found in, kind of in the inner part of your cerebrum, maybe in the medulla region of your cerebrum. But either way, it has a couple of structures that are important. So when you process emotion, fear, happiness, whatever, most of your emotions are going to be processed in this way. And this is the limbic system, and that's what they've discovered. But then we can break that down a little more, in a little more detail, and we see things like the amygdala. Okay, so this is an important structure because we kind of know a lot about it, and there's been a lot of study, a lot of research done on the amygdala, but it's associated with threats, anger, but really it's kind of fear and the response to fear. They've done studies that show that people with a larger amygdala tend to feel fear more and consider things a threat more. I don't know what's been done with that study since I read about it. So that's the amygdala, and then the hippocampus is involved in memory. And memory is, of course, I have down here, memory is strongly tied to emotion, or emotion is strongly tied to memory. So if something is considered salient or important, if something is important, if we have an emotional response to something, we're more likely to remember it. So that's kind of how it's tied to memory. If you just go through your day-to-day routine and nothing exciting happens, it's going to fade from your memory more quickly than if something scares you or makes you happy, or just kind of gets you excited. Well, I don't know, makes you sad. Then those things tend to be stored as memories. So other regions, the hypothalamus kind of gets into it. The thalamus for that relay center. The basal ganglia, which is, like I said, involved in movement, but there are some structures like the reward and things like that are also, remember that there's a substantia nigra, and there's some other areas, nucleus accumbens, that are involved with reward and gratification that can also be considered part of the limbic system. Olfactory cortex, cingulate gyrus, which can give, kind of provides some of that meaning to emotional things. But anyway, they all kind of work together, and together they're called the limbic system. All right, so let's see. I think that's it. So this is just a table that kind of shows the different regions and kind of has some additional information on them. But that's the last slide.